Applied Human Biology, Unit 1, C2 Genetic Disorders. So some genetic terms first of all. So a gene is a sequence of DNA that codes for a particular polypeptide. An allele is a different form of a gene. A phenotype is an organism's physical appearance or visible traits. Genotype is the genetic makeup of an organism. Heterozygous, an organism that has two different alleles for a trait. Homozygous, two alleles are the same. Dominant is an allele that is always expressed. Recessive is only expressed in the homozygous state. Sex linkage is the presence of a gene on a sex chromosome, usually the X chromosome. A carrier is one of the two alleles are carried by this person, and it's the recessive allele for the disorder that they carry. Affected or sufferer is the diseased phenotype. The non-affected or non-sufferer is a non-diseased phenotype. Genetic diagrams show the possible genes of the offspring. A Punnett square shows all the possible outcomes of a genetic cross. Familial pedigrees can be used to show and record the inheritance of genetic disorders in a family tree. So this is a genetic diagram. A Punnett square is one like this, where you cross the alleles together. Weaknesses of genetic pedigree diagrams, so these ones. The diagram does not definitively show who are the carriers. It assumes that the family report the information accurately. Other methods such as genetic screening would give a more definite result. Haemophilia is a sex-linked inheritable disease characterised by the loss of impairment of the normal clotting ability of the blood, so that a minor wound may result in fatal bleeding. So now looking at specific types of genetic disorders. So cystic fibrosis is caused by the recessive allele, so we usually show it as a little c. And usually people can inherit it if a mother had a baby with a father that they are both carriers. So they're not actually showing the symptoms, but they are carriers of the disorder. And then they have a 1 in 4 chance of having a child with cystic fibrosis. Symptoms of cystic fibrosis: the mucus becomes thicker and stickier and build up in some internal organs, leading to multiple pleiotropic, which means that one gene influences lots of unrelated phenotypic traits. So it's got a pleiotropic effect. Abnormal absorption of nutrients in the small intestine, chronic bronchitis, etc. Cause of cystic fibrosis is a mutation of the CFTR gene on chromosome 7, which causes the chloride ion channels to not function, so chlorine cannot pass out of the cells properly. Huntington's disease. Huntington's is caused by a dominant allele this time. Cause of Huntington's disease is a gene affecting neurological function and means a lack of neurotransmitter called GABA is produced. Symptoms of Huntington's disease are tremors, clumsiness, memory loss, inability to concentrate and mood changes. So the thing with Huntington's is it usually is late onset, so they don't develop the symptoms until later in life. By that time they will already have passed on their genes. So if, for instance, a male that was heterozygote and had the dominant allele had a child with a female that wasn't a carrier or anything, then they would have a 50% chance of their child developing Huntington's disorder. But it may be that this person didn't know they had Huntington's because it's a late onset disease. Haemophilia is caused by a sex-linked recessive allele. So you would usually show, obviously the male is XY and the female is XX, so the male only carries one version of that particular allele. And the reason things like this are more common in males is because they don't have a corresponding allele to counteract the recessive one. The symptoms of haemophilia are bleeding of the joints, gingival bleeding, unable to form clots at wounds, The causes of haemophilia are a lack of blood clotting factor or low numbers of blood clotting factors. Remember that the male genotype is XY and the female genotype is XX. Down syndrome is a congenital disorder caused by having an extra chromosome 21, and that's known as trisomy, and it's caused by chromosome non-disjunction during meiosis. Symptoms of Down syndrome are physical growth delays, intellectual disabilities, facial features, heart and circulatory problems, and bad immune systems. The risk factors of Down syndrome is advanced maternal age. I think it's over the age of 38, the risk increases quite significantly. Already having a child with this Down syndrome, being a genetic carrier of the genetic translocation, and Down syndrome diagnosis is through ultrasonography in utero or amniocentesis or chorionic villus sampling. Cancer is a disorder in which some of the body's cells lose the ability to control growth and divide uncontrollably. Metastasis is the spread of cancer cells beyond their original site. A benign tumour is an abnormal mass of cells that remains at its original site in the body. A malignant tumour is an abnormal tissue mass that can spread into neighbouring tissue and to other parts of the body. So that would be a cancerous tumour. Proto-oncogenes, normal cellular genes that regulate cell division. A tumour suppressor gene slows cell division. So an oncogene is a mutation of this proto-oncogene, which is a cancer-causing gene formed because of mutations. And then mutation of the tumour suppressor gene means that it allows unregulated cellular growth. So a tumour suppressor gene, normally it regulates, it slows down cell division, but if that gets mutated, then the cells can divide uncontrollably. Proto-oncogenes, they normally regulate cell growth, but then if they mutate into oncogenes, then it means that it can, again, there's not as much cell death and it will just replicate quite fast. So testing now, so DNA sample source comes from saliva, blood, semen and mucus. Limitations of DNA samples is that chemicals used to prevent clotting may, when taking a DNA sample, can affect the quality of the DNA. May require multiple blood samples from more than one family member for comparison. Advantages of genetic testing: increased knowledge about your risk of developing a disorder, information to help make informed medical and lifestyle decisions, can educate other family members, earlier detection, which increases the chance of survival. Disadvantages of genetic testing: testing may increase anxiety and stress. Testing does not eliminate a person's risk. Cancer results in some cases may return inconclusive or uncertain, increasing insurance premiums and genetic discrimination. Genetic tests during pregnancy. So tests can be conducted while the fetus is in utero to detect genetic conditions. A blood test is carried out to assess the level of risk of the fetus having a genetic disorder before amniocentesis or chorionic villus sampling takes place, as there is a small risk of miscarriage with these tests. So chorionic villus sampling takes cells or DNA from the placenta, which have the same DNA as the fetus, and it allows earlier testing than amniocentesis from 11 to 14 weeks. It allows for genetic testing of chromosomal disorders. Tests must be followed up by a blood test afterwards, and you can also tell the sex of the fetus earlier than observed with ultrasound. Amniocentesis: the needle is inserted through the abdomen and takes a sample of amniotic fluid to obtain fetal cells with their DNA. Testing is done when the fetus is more developed, from 15 to 20 weeks. You can test for genetic or chromosomal disorders, provides information about neural tube defects, provides information about chromosomal abnormalities like Down syndrome, Edwards syndrome and Pateau syndrome. Disadvantages of chorionic villus sampling and amniocentesis: they increase the risk of miscarriage with both tests, and the risk is about the same for both. There's a risk of infection in both, risk of sensitization or rhesus disease, if the baby is rhesus positive and the mother is rhesus negative. Discomfort during and cramps after the procedure, not offered to all pregnant women, only people that have a higher risk. Women may be more likely to opt for testing if there is a history of genetic disorders in the family. Some people may be ethically opposed to prenatal testing. Prenatal testing is optional and does not need to be accepted, even if it's offered. Tests can't check for every single potential disorder. Disorders tested for may depend on the family history or the lab that performs the test.